while we have discussed in great detail how the fuel in the form of reduced uh, electron carriers are burned in the respiratory chain to produce the proton gradient, the question remains to be answered how it is actually fueled. And one obvious way is through citric acid cycle that runs in the mitochondrial matrix and generates this reducing equivalents NADH and FADH2, which could be directly fed from the mitochondrial matrix side into respiratory chain through complex 1 and 2 respectively, as we discussed previously. However, how to deal with the catabolic processes that take place outside the mitochondrial matrix in the cytoplasm, and one important process is glycolysis that takes place in the cytosol and generate constant flux of NADH, which has to be um, uh, regenerated back into NAD plus to run next cycles of glycolysis. So there must be a way to transfer NADH from the uh, mitochondrial cytosol to mitochondrial matrix to burn it into respiratory chain. And one variant would be to directly transfer NADH through some channels into mitochondrial matrix. However, it doesn't solve the problem of regenerating NAD+. If NADH would be transferred, the NAD+, which is necessary for glycolysis, would be depleted. So there must be some kind of indirect way to um, transfer the electrons, the reduced equivalents, in a clean manner from cytosol to mitochondrial matrix. And there are two systems which implement in our cells. One is called malate aspartate shuttle, and another is called glycerol 3-phosphate shuttle. Malate aspartate shuttle and glycerol 3-phosphate shuttle are prevalent, have different prevalence in different tissues, while malate aspartate is prevalent in tissues like liver, heart, and kidney. Glycerol 3-phosphate shuttle is prevalent in brain, for example, and muscle tissue. So let's look at these two systems of transfer and reducing equivalents in more detail. The malate aspartate shuttle, we did touch upon its elements when we discussed gluconeogenesis and discussed the way how the pyruvate can be channeled through mitochondrial matrix to convert it to oxaloacetate, combination of oxaloacetate and NADH, which are vital for running gluconeogenesis. And components of the same system are used to recycle the transfer reducing equivalents from cytosol to mitochondrial matrix when this oxidative, when the catabolic fates of glucose are prevalent. So, so essential component of the primary component of the system are these two twin malodehydrogenase enzymes. One present in the cytosol and another present in the mitochondrial matrix. In malodehydrogenase in the mitochondrial in the cytosol, sorry, takes oxaloacetate and uses NADH generated, for example, during glycolysis to reduce oxaloacetate to malate and concomitantly produce NAD plus oxidized form, which could be used for running next cycles of catabolic processes. And then there are channels which we also briefly touched during gluconeogenesis discussion that can transport malate into mitochondrial matrix, and the twin of the malate dehydrogenase in the mitochondrial matrix performs the same reaction but in opposite direction, taking malate and NAD plus within mitochondrial matrix, producing NADH and forming oxaloacetate. And this solves the problem of transfer in a clean manner electrons, reducing equivalence from the cytosol to mitochondrial matrix. There is no net change in NAD as a electron carrier, but it's just this system just changes the balance of NAD plus between the cytosol and the mitochondrial matrix pumping NADH um, electrons converting NADH through the membrane. However, while solving this problem, cells create a problem where oxaloacetate has to be constantly evacuated from the mitochondria, or sorry, from the cytosol, and would constantly accumulate in the mitochondrial matrix. So to balance this cycle, cell must somehow have a way to evacuate oxaloacetate from the mitochondrial matrix and pump it back into the cytosol to run this transfer of reducing equivalents over and over. And this is done by transaminating of oxaloacetate and producing a molecule called aspartate, which is amino acid and essentially the same as oxaloacetate, but the keta group is changed to amino group, and this is done by the mitochondrial enzyme called aspartate aminotransferase. And there are channels that can transfer aspartate through the inner mitochondrial membrane and channel it back to the cytosol. And there is a twin enzyme, like in the case of malodehydrogenase, the cytosolic aspartate aminotransferase that exchanges amino group in aspartate back to keta groups, thus regenerating oxaloacetate. So in this way, the whole cycle could be completed, and hence this the name of this system that used to flow reducing equivalents to the mitochondrial matrix malate aspartate shuttle. However, if you look at finer details, while solving this problem of recycling oxaloacetate, the cell generate a problem of disbalance in amino groups. So the amino group is transferred in the mitochondrial matrix and have to be released in the cytosol. So there will be net flux of amino group in the matrix to cytosol direction. So the cell must also solve this problem of how to regenerate back the flux of amino groups. And this is done through the coupled shuttling of the in the amino group transfer factors, combination of glutamate and alpha ketoglutarate. Glutamate and alpha ketoglutarate is kind of the same pair as aspartate and oxaloacetate, but having one more methylene group in the backbone. So, and aspartate aminotransferase, which transfers amino group from oxaloacetate to aspartate, uses as a donor in the mitochondrial matrix amino acid glutamate, and then concomitantly producing oxaloacetate, which is the same pair, but where amino group is changed to keta group. Likewise, the in the cytosol, the twin enzyme uses this pair, but in the opposite direction, to transfer the amino group backwards. And the catch is that these channels transferring malate and transferring aspartate at the same time transfer the alpha ketoglutarate and glutamate in the opposite direction, so they act as antiporter. Malate channel is actually antiporter that couples transfer malate into one direction with the transferring of alpha ketoglutarate in the opposite direction, and the channel which transfers malate in one direction is actually anteported at the same time transfer glutamate in the opposite direction. This way the flux of the amino group is restored. So the net product of all this complex shuttle is that the reducing equivalents are transferred from the cytosolic location to the mitochondrial matrix location. So, and this is a very efficient way to fuel the mitochondrial electron transport chain from the catabolic process that generate reducing equivalents in the cytosol. So the complementary system which functions in these other tissues, glycerol 3-phosphate shuttle, is much more simple. During glycolysis, the NADH, which is produced during the oxidative phosphorylation step, is recycled to NAD plus and is reducing the yet another product of glycolysis, the hydroxyacetone phosphate into glycerol 3-phosphate, and this is hence the name of this mechanism to feed reducing equivalence glycerol 3-phosphate shuttle. The glycerol 3-phosphate is in turn oxidized back to hydroxyacetone phosphate by the glycerol 3-phosphate dehydrogenase, the membrane-associated protein which is associated with inner mitochondrial membrane. So the this enzyme used FAD as cofactor that is concomitantly reduced to FADH2, and unlike the malate aspartate shuttle, the glycerol 3-phosphate shuttle cannot feed the electrons into the mitochondrial matrix, and it acts very similar to complex 2 by using FADH2 to reduce ubiquinone, which is floating within the lipid phase of the inner mitochondrial membrane that is straight fed to complex 3. So this way the NADH fed into respiratory chain through glycerol phosphate shuttle produces less proton pumping as compared to malate aspartate shuttle. And this is it regarding the feeding pathways of respiratory chain.